Hello, welcome to Module 5, Chapter 9, talking about muscles and muscle tissues. This is Part 2, the sarcomere. So in this cartoon, you can see the skeletal muscle details here. When you look at a H&E stain for skeletal muscles, you can see these if you look close, you can see dark bands and light bands. And the same thing in cardiac muscle over here. You can see a dark band and a light band, a dark band and a light band. And that's what we're going to talk about today. What structures are making up those dark and light bands? So if we look at the top right-hand corner of this slide, I've written a muscle. So M dot or M period, that means muscle. So I'm going to abbreviate that. Muscle fiber or a muscle cell. So remember, we talked about this in the last lecture. You have the epimysium, and then the paramysium and the myomysium. So the myomysium, or excuse me, the endomysium. So on the outside is the epimysium, and then the mysium that surrounds each one of these fascicles. It's called the paramysium. And then when the paramysium dives in here and surrounds a single muscle cell, that's called the endomysium. And so if you take one of these single muscle cells here and you enlarge it, it looks like this cartoon. So this is a single muscle cell. This purple thing is a nucleus. Here's another nucleus. So remember, skeletal muscle cells are multinucleate. And then this thing that's cut and pulled back, this white thing, that's the plasma membrane, aka the plasmalemma. And in muscle physiology, they call the plasma membrane or the plasmalemma, they call it the sarcolemma. And you can see inside a single muscle cell, it's full of these things called myofibrils. And then you can also see these orange things here. Those, of course, are mitochondria that make ATP to fuel your muscle cells. So, and then you can see in the myofibril, you've got these dark bands, and then you have these light bands with a line through them. So we're going to talk about those today. So the sarcomere is the functional contractile unit of a muscle cell, and it's defined as the region of a myofibril. So again, this is a single myofibril, and you can see there's hundreds of myofibrils in a single muscle cell. So the sarcomere is the region of the myofibril contained between two Z discs. So this is a Z disc, and this is a Z disc. So between those two Z discs, you would have a single sarcomere. And in the part of the cartoon that shows the myofibril being pulled out of the muscle cell, you can see there's 1, 2, 3, 4, 5, 6, 7, 8, 9 sarcomeres showing in the cartoon. Skeletal and cardiac muscle cells have sarcomeres, but smooth muscle cells do not have sarcomeres. So if you pull out one of those myofibrils from inside a skeletal muscle cell and you enlarge it, you can see the I band is here. So this is the I band. Remember, that was the light band, and then the A band is the dark band. So this part here would be the dark band. So in the early muscle histologists saw this, they saw a dark band and a light band, and they named them that. So inside a I band or a light band, you have this structure called a Z disc. And inside the A band, you have a zone called the H zone in the middle of the A band. And then in the middle of the H zone, you have this structure called an M line. So a lot of terminology with muscle physiology. So those are zones, these discs and zones and bands. Those are all just superficial structures, but the proteins that make up all these discs and zones and bands are actin, the protein actin, and the protein myosin. So these blue lines are called thin filaments, and they're made of the protein actin. And then these orange lines are called the thick filaments, and they're made of the protein myosin. And then I asked the question, in what region of the sarcomere do actin and myosin overlap? So you have to find a sarcomere that goes from Z disc to Z disc. So this whole region would be a sarcomere. So where do actin and myosin overlap? So you can see here's an actin protein. Here's a myosin protein, and they are overlapping each other in the A band. So the answer would be the A band. So here's a different cartoon that talks about the same thing. So here's this is the muscle fiber or the cell. It's cut, and you look inside the muscle cell, and you see that it's packed with these things called myofibrils. And then the myofibrils are made up these successive functional units called sarcomeres. So remember, sarcomere goes from Z line to Z line. And then in this cartoon, it shows what makes up a sarcomere are these thick and thin filaments. So the thick filaments are in red here, and they're made up of myosin, and the thin filaments are in blue, and they're made up of actin. And in this cartoon, it gives you a little bit more detail because it shows that the thick filaments made of myosin have these little cross bridges that are linking to the protein actin in the thin filament. So this is a different cartoon of the sarcomere. It's a little more detailed. So here's the sarcomere. Remember, sarcomere goes from Z disc to Z disc, and you have the A band, which is the dark band, which is where myosin and actin overlap, and then they have the light band over here that just has the actin or the thin myofilaments in it. This shows a little bit more level of detail because it shows myosin, but now it's not just a single flat rod. It's got these little myosin heads. So all these little myosin heads are on myosin, and then these myosin heads will actually bind to myosin head binding sites on actin, this blue line here. And this also shows a little more detail how the actin is sort of, what's the word, how it's anchored, how the actin is anchored in the Z disc. So let's see. Oh, yes, I told you that. The myosin head form cross bridges when they link to myosin head binding sites on actin. The myosin heads also contain these things called ATPase enzymes. So the myosin heads can move when they bind ATP and burn the ATP. The heads will move, so they'll attach and detach from the myosin head binding sites on actin. So we talked about this. The thin filaments are in blue, and they're composed of the contractile protein called actin. Now in this cartoon, we show over here on the left, so here's the skeletal muscle fiber, aka a cell. It's made up of a whole bunch of myofibrils, and then it enlarges the myofibril over here in the middle of the screen. And we've talked about these bands and zones. Now when we go, and also here's the sarcolemma, so we go deep to the sarcolemma. Here's mitochondria, so you see mitochondria throughout the skeletal muscle cell. But now we're going to look at a new structure that sits on top of the sarcomere, and that is the sarcoplasmic reticulum, so that, or the SR. That's all this blue stuff. This is all SR. It's all blue stuff, and it's contains calcium. And then, so the SR, when it gets thick here, so here, when the SR gets thick, that's called a terminal cistern, and it's linked up with this thing, this white thing called the T-tubule. So when an electrical impulse comes down into the T-tubule, it will connect with the terminal cistern of the SR and allow for the terminal cisterns of the SR to release calcium. And when calcium is present, you can have the myosin heads forming cross bridges with actin and making the sarcomere get shorter. And when you have millions of sarcomeres getting shorter along the length of a muscle, you can contract a whole muscle. Okay, and we talked about that. So the sliding filament theory then that allows for muscle contraction, like I said, when you have, you know, thousands or hundreds of thousands of these sarcomeres all getting shorter at the same time, that whole muscle cell will get shorter and you will contract. So the sliding filament theory says that when these myosin heads bind to myosin head binding sites on actin, and then the ATP is burned and the myosin heads can move, the myosin heads can walk along the actin, and when they do that, the whole sarcomere gets shorter, so the two Z discs get closer to each other. And we'll have another lecture or video on the sliding filament theory, but for now, you can get your Zen candles out and you can imagine as if these little myosin heads walking along the actin myofilament, making the sarcomere get shorter and shorter, thus contracting a whole muscle. And that's it.